on that. So what does it mean? That is, if you have very simply capital A, simple B, white flowers. You don't have a capital B dominant allele to help us produce proteins. If you have capital simple A, simple B, still what happens? We don't have we don't have a capital B dominant allele, so we can't produce purple flowers. So that's the story behind this. Hope you understood that clearly. I'm taking a lot of time on genetics, I feel, probably because when I explain, I like to explain logically, to see the logical sides, because it's no point of memorizing these definitions. It's not like other lessons, you have to understand explicitly the definitions. Inheritance of a particular phenotype. So what's the next definition we are moving on to? Polygenic inheritance. So what do you mean by polygenic inheritance? Inheritance of a particular phenotype, such as qualitative characters. What are those qualitative characters? Are they qualitative characters? No, quantitative characters. Sorry, so inheritance of a particular of a phenotype, such as quantitative characters. What are those quantitative characters? Height, you can measure them, right? The height, you can measure the height. The skin color, intelligence quotient, which results from a cumulative expression, a combined, let me, cumulative means combined expression of two or more genes. So inheritance, once again, of a phenotype, such as quantitative characters. Examples: height, skin color, intelligence quotient, which results from a cumulative expression of two or more genes, is called polygenic inheritance. See, from the word itself you can get the meaning, right? Polygenic, many genes are involved. So by the cumulative expression, the combined expression of two or more genes, right, this is polygenic inheritance. So that's a very easy definition. We'll move on to the next definition. So I'll give a little explanation. I feel bad for not explaining a bit more about it. So for example, when you take the skin color in humans, it is determined by many genes. For simplicity, only 3 genes are considered, right? Now each gene, capital A, capital B, or capital C, has a dark skin allele, which is contributing 1 unit of darkness to the phenotype and being incompletely dominant to the other allele. So it's, remember the word incompletely dominant. It's not perfectly dominant. It is incompletely dominant to the other allele. That is simple A, simple B, or simple C. Therefore, if you take a person like this, right? I'll show you that. If you take a person like this, capital A, capital A, capital B, capital B, capital C, another person, simple A, simple A, simple B, simple B, simple C, capital A, simple A, capital B, simple B, capital C, simple C. What can you say about these 3 people? The first person has a very dark skin. Second person has a very light skin. Third person is intermediate between the skin color. It's very obvious, right? Because each gene, capital A, capital B, or capital C, has a dark skin allele. This only has dark skin alleles, right? The first one. The second one doesn't have any dark skin alleles, so it should be very light skinned. But the third one has dark skinned alleles. Then they are in dominant conditions. but they are not perfectly dominant, right? See the definition that we were talking about, they are not perfectly dominant. So that's why this fellow number 3 gets an intermediate color in between dark and fair skinned. So it's kind of brown. So once again, let's see the definition quickly. Inheritance of a phenotype, such as quantitative characters, what, height, skin color, intelligence quotient, which results from the cumulative expression of 2 or more genes, as we saw in our examples, is called polygenic inheritance. That's perfect. Now let's go to the next one. What is it? Got to see what the definition is. That's a big, long one. Oh my goodness, 10 sentences, 10 points actually. So I can't be underlining because I want to show you the whole definition. So let's see anyway. So what is the definition? What is genetic linkage? Big definition. What is genetic linkage? Some genes coding, right? Some genes coding for particular characteristics. Some genes coding for particular characteristics are located on the same chromosome, right? So some genes coding for particular characteristics are located on the same chromosome and also at a closer distance. It's surprising, right? Some genes coding for particular characteristics, different characteristics, are located on the same chromosome though, and also at a closer distance added to that. At a closer distance added to that, thus escaping from crossing over and independent assortment occurring during meiotic cell division. At gametogenesis, and what happens is that finally they inherit together as unit, resulting in a deviation from Mendel's law of independent assortment. The above phenomena is called genetic linkage. So don't be afraid of this definition. Try to understand it. Let's say we can all the time. Always have this positive energy. I know these things. I can. It's very easy for me. Other people have said that it's complicated, right? No, it's not complicated. It may be complicated for them, but for me it's very easy. Think like that. So some genes coding for particular characteristics are located on the same chromosome and also at a closer distance, thus escaping from what we call crossing over and independent assortments occurring during meiotic cell division at gametogenesis and inherit together, resulting in a deviation from Mendel's law of independent assortment. The above phenomena is called genetic linkage. Now I think I should explain a bit more about it. Now normally what happens is that, let's say we have the homozygous wild type, the flies, right? Gray-bodied, normal winged flies. Capital G, capital G, capital G, capital N is crossed with, who's the next one? We have simple G, simple G, simple N, simple N. So who is this organism? Homozygous wild type organism, which is gray-bodied and normal winged. The next one is a double mutant, which is black-bodied and has vestigial wings. Now they are doing, let's do this according to Mendel. Let's say Mendel did not know about the experimental results, and as usual, let's say we gave him a problem to solve. He's not living now, but let's say we gave him the problem to solve according to his methods. Right, he's saying, okay, well, this is the parental generation, right? He's saying parental generation, and he says, oh, okay, give me the gametes. Capital G, capital N, and we have simple G, simple N, sorry. Then we have an organism, capital G, simple G, capital N, simple N. So far is correct. No problems with that. and we have 100% gray-bodied, normal-winged flies. Now comes the problem. Now Mendel says, right? Mendel is saying capital G. Okay, so simple. Why are you giving me so? Why are you giving these kind of questions to me? This is too simple for me, he says, right? So we are doing a test cross now. You know what a test cross is? I gave you the definitions. Now, what kind of gametes can you form? Capital G can go with capital N. It can go with simple N. Simple G can go with capital N. Simple G can go with simple N. So we get capital G, capital N. We get capital G, simple N. It's not very clear. Sorry for that. Simple G, capital N. We have simple G, simple N. And then from the other side, we have simple G, simple N, 100%, right? So you see, this 25%, these gametes. What do you get at the end? I'm not trying to show all those. You know how to join these things and get. You get capital G, capital N, simple N, gray-bodied, normal. You get capital G, simple N, gray-bodied, vestigial wings. You get simple G, capital N, simple N, black-bodied, normal wings. You get simple G, simple N, black-bodied, vestigial wings, right? So you have the genotype ratio of 1:1:1:1, and you have the phenotypic ratio, which is equal to genotypic ratio of 1:1:1:1. Now I'm coming to Mendel and saying, sir, right, but in our experimental results we got something completely different. Now he's surprised. Can't be, because I used my principles and it turned out to have these answers. And then with time, right, let's say I discovered the reason. I discovered this problem is actually due to genetic linkage, which Mendel has never seen before. Alright, now let's forget about Mendel and this come to our era now. Right, we can see then. Now, according to the new findings, what we see, we have capital G. Sorry, so we saw this before. We have capital G. So that's too ugly. We have capital G, capital N, crossed with simple G. I'm not going to tell the names because I don't have much time. Right, we get that back again. Capital G, capital N, homozygous wild type, gray bodied, normal wings, crossed with simple G, simple N. So now the surprising factor that we find out is that capital G and capital N love to stay together. They stay as a unit. Oh my god, see the love affair. They're together. And the other one, they're also together. The simple G and the simple N are together. Now when these gametes fuse in the F1 generation, we have in disguise capital G, simple N, capital G, capital N, and we have simple G, simple N, right? Now, when you see the F1 generation, you don't see any difference. We have gray-bodied normal wings, and we saw in the Mendel's way also that we came across this. But now, when these alleles segregate, when you're coming to the F2 generation, when you cross the F1 generation, when you do a test cross to the F1 generation, you see something completely different, right? What's that? What you see is this. Now, in the F1 generation we got, right? What do we get? We get capital G, capital N, simple G, simple N, right? Hope it's clear. Now, this is the organism. Then we have to do a test cross. capital G, I'm sorry, simple G, simple N, simple G, simple N. Now what happens when you produce gametes? You have this same type, simple G, simple N, and you have what? Two different types, right? Capital G, capital G, capital N, simple G, simple N. Now what happens? What happens if you cross? What happens when these gametes fuse? We get, I show it in the next page. So remember this slide. Then we come to the next one. What do we get? We get capital G, capital N, simple G, simple N. We get simple G, simple N, simple G, simple N. Do this in your papers and see. Now these organisms in the F2, the genotypic ratio of G, capital G, simple G, capital N, simple N is to simple G, simple G, simple N is 1:1. So you see, earlier we found 4 types of organisms resulted, right? But now only 2. Why is that? Because you saw that now, right? You saw. So go back to my how I did the cross and you find out, right? Because now capital G and the capital N are together as a unit, and simple G and simple N are together as a unit. So for example, like this, they can get together, right? Capital G, capital N can go with simple G, simple N. They can get together and form this capital G, capital N. So these two gametes are in one cell now, right? Or it can be from the gametes that we produce, simple G, simple N can go with simple G, simple N, right? Look at the resource book again and try to understand that better. So I did all I can. It's very inconvenient with these small phones, very difficult to do. I can't show you the entire screen. So anyway, I hope you understood that. So let's shout out this definition together. Some genes, right? Some genes coding for particular characteristics are located on the same chromosome and also at a closer distance, thus escaping from crossing over, and independent assortment occurring during meiotic cell division. Of gametogenesis inherit together, resulting from the deviation from Mendel's independent assortment. We saw that, right? The above phenomena is called genetic linkage. I can explain more and more by taking the cells and things like that, and then the meiosis, the process, but it's going to take a long time. So I hope you all can understand that through the books. So I explained as much as I can about gene linkage, because they're closely together. They escape from what, right? They escape from independent assortment and crossing over. Now let's go through the other definitions very quickly. Red-green color blindness is an X-linked what? Is an X-linked. So is this recording? Yeah, it is. So red-green color blindness, what is it? Is an X-linked recessive disorder characterized by the difficulty in perceiving differences between red and green color. So we have a problem with red and green color perception. We can't differentiate between those two colors. So red-green color blindness is an X-linked recessive disorder. Remember that it's X-linked and it is a recessive disorder characterized by the difficulty in perceiving differences between the red and the green colors. So what are X-linked ones? Let's see. You know what X-linked characters are? Characters expressed by or carried on the X chromosome are called X-linked characters. And the genes expressing or carrying those characters are called X-linked genes. So once again, characters expressed by or those that are carrying or those that are being carried on the X chromosome are called X-linked characters, and the genes expressing or carrying those characters are called X-linked genes, right? Simple. Now, on the other hand, for the other definitions also, the characters which are expressed by or carried on the Y chromosome are called Y-linked genes, and the genes expressing or carried by those characters are called Y-linked genes. So once again, red-green color blindness is an X-linked recessive disorder. This time it's a recessive disorder characterized by the difficulty in perceiving differences between red and green color. Now let's not take a long time because these definitions are very short. We discussed the great difficult details of the definitions that were hard. Now we'll do it faster because we have to complete a lot of definitions. Right, the next one is very important. There are more important, yeah, several important definitions, so everything is important here. The next one, an X-linked recessive disorder where one or more proteins required for blood clotting are absent. So what is that? I forgot to write that. It's hemophilia, right? Hemophilia. So remember, what is hemophilia? An X-linked, right? Hemophilia is an X-linked. It's also X-linked, you see, disorder where one or more proteins required for blood clotting are absent. So you see, it's a X-linked recessive disorder. Remember that first, and then you have to remember that where one or more proteins required for blood clotting are absent, because those proteins are absent because the genes are not functioning, maybe, right? Genes that make these proteins. So an X-linked recessive disorder where one or more proteins required for blood clotting are absent. So what is pleiotrophy? Pleiotrophy is the phenomenon where expression of a single gene affects the expression of multiple traits which are not related to each other. Pleiotrophy is the phenomenon where expression of a single gene, just the expression of one gene, affects the expression of multiple traits, different features, which are not related even to each other, right? Wonderful thing happening, right? Pleiotrophy is the phenomenon where expression of a single gene, when that gene is expressed, it affects the expression of multiple traits which are not even related to each other. So let's come quickly to the other definitions. So what is sickle cell disease? Sickle cell disease is a disease caused by an alternation in the hemoglobin protein. Sickle cell disease is a disease caused by the alternation in the hemoglobin protein, where in the red blood cells, right? So sickle cell disease is caused due to an alternation in the hemoglobin protein of red blood cells. Now you will see the greater details of this in molecular biology. In that they are mentioned about the gene sites behind this, right? What kind of mutations happen. Better refer that again. Don't take a long time for that. So it's in another lesson. But also in genetics also they have mentioned about sickle cell disease. But greater details are in molecular biology and the mutations. So sickle cell disease is a disease caused by an alternation in the hemoglobin protein. So when that hemoglobin protein is changed, right? Alternated. This sickness is resulted. Those are very short and simple definitions. I don't have to give a great detail about those things. The next one is this. What is cystic fibrosis? Now, cystic fibrosis is a disease condition causing what? It causes thicker and stickier mucus. So thicker. The mucus is thick, feature number 1, and the mucus is also stickier, feature number 2. Then its normal nature. Normally you don't find, normally it's let's say sticky to some extent, let's say stick to some extent, but it's stickier, it's thicker than the normal mucus, which is an autosomal. Remember that it's an autosomal recessive disorder. Autosomal recessive disorder. Actually, if you are gonna have, I'm not saying you all are gonna have that, but if you are going to have an autosomal recessive disorder, you have to inherit two mutated genes, one from each parent. Now, these disorders are usually passed on by two carriers, right? So you don't have to know much detail about this. Just remember that. So cystic fibrosis is a disease condition causing thicker and stickier mucus than its normal nature, and which is an autosomal recessive disorder. The meaning of autosomal really is actually that the gene is located not on a sex chromosome, it's on a different chromosome. So that's the meaning. So the next one, now I hope you understood that. Let's go to the next definition. And we'll stop from Hardy-Weinberg's definition. The study of occurrence of certain phenotypes. Study of occurrence of certain phenotypes. Study of the occurrence of certain phenotypes of certain characters controlled by factors other than DNA sequence or genetic code is called epigenetics. So what is epigenetics? It is the study of occurrence of certain phenotypes of certain characters controlled by factors other than their DNA sequence or the genetic code. So other than the DNA sequence and the genetic code, due to several other factors, right? They're still studying about it due to several other factors, except for those DNA sequence or genetic code, right? Certain phenotypes of certain characters are controlled. You see, study of occurrence of certain phenotypes of certain characters controlled by factors other than their DNA sequence or their genetic code is called epigenetics. Apart from the genes, epigenetics, beyond genetics, right? Something else. All right, so then the next definition, now we'll read that once again first. Epigenetics means a study of occurrence of certain phenotypes of certain characters controlled by factors other than their DNA sequence or genetic code is called epigenetics. So what is epigenetic inheritance? Inheriting epigenetic traits from parents to the children's generation is called epigenetic inheritance. So inheriting epigenetic traits from parents to the children's generation is called epigenetic inheritance. So see, they can be inherited also, epigenetic traits. So inheriting epigenetic traits from parents to the children's generation is called epigenetic inheritance. So what is Hardy-Weinberg's equilibrium principle? A nice one. Hardy-Weinberg's equilibrium principle, frequently asked in past papers, a popular definition. In a population that is not evolving, in a population that does not evolve, allele and genotype frequencies will remain constant from generation to generation. So in a population that is not evolving, the allele and the genotype frequencies will remain constant, unchanged, from generation to generation. This is known as Hardy-Weinberg's equilibrium principle. I think you remember about, you have done many calculations from this. So just remember the definition, right? Remember the calculations that you have done. Obviously, if you have done the calculations, you can easily remember the definition. You know that famous equilibrium of Hardy-Weinberg, p squared, p squared plus 2pq plus q squared equals 1. What we saw earlier is that, what did we see? The sum of the frequencies of all three genotypes, right? We are taking a sum, right? Sum of these frequencies of all three of these genotypes is equal to 1. So we can see from this also whether population is evolving or not and things like that. So let's end it from here.